Good morning and welcome to those in the auditorium and online to the Doherty Institute Vaccine Media Conference. My name is Professor Sharon Lewin and I'm Director of the Doherty Institute. And I'm speaking to you from Melbourne, and I'd like to start by acknowledging the traditional owners of the lands upon where we are, the Wurundjeri people of the Kulin Nation, and pay my respect to their elders past and present. So this week, we were extraordinarily lucky to have Professor Dame Sarah Gilbert visit the Doherty Institute as our guest for our annual symposium. The Doherty turned 9 years old earlier in September, and we celebrate that event each year with a symposium. And this year, the theme was around vaccine development for SARS-CoV-2, and who else could give us the most brilliant overview of how a vaccine was developed by an academic institution and ultimately licensed and given to millions of people than Dame Sarah. So Sarah's story of how the Oxford AstraZeneca vaccine was developed is an extraordinary one. And at the same time, we also had the great outcome of developing a vaccine here at the Doherty Institute. And that's what we'll be speaking about, together also with other efforts across the country around developing COVID vaccines. More importantly, we're all thinking about how we can do this better, faster, and in a more collaborative fashion for future pandemic challenges. So before I come to ask our panelists questions and then open it up to those of you in the auditorium and online, I will introduce our panelists formally. I'd also like to note that Rebecca Elliott, Head of Communications at the Doherty Institute, is here with me, and Rebecca will facilitate the questions from you who are joining us online. So, Professor Dame Sarah Gilbert needs no introduction, but formally, she is the Said Chair of Vaccinology at Oxford University, and in 2021 became the head of the Outbreak Pathogens Vaccine Group in the Pandemic Sciences Institute at Oxford. The Pandemic Sciences Institute is also newly established in response to the pandemic and is a very interesting initiative that we've spent a lot of time discussing over the last few days with Sarah's visit. In 2020, Professor Gilbert initiated and led the rapid production and development of the ChAdOx vaccine, or Vaxzevria, not good on that one, which was ultimately licensed to AstraZeneca and now in use over 180 countries, and of course widely used in Australia following manufacturing by CSL. Professor Gilbert was made a Dame Commander of the British Empire in 2021 in recognition of her work during the pandemic. And I'll also note her book, highly recommended read, Vaxxers, describes the development of the ChAdOx vaccine, was a Sunday Times bestseller and is a brilliant book. Professor Terry Nolan, AO, is the University of Melbourne Redmond Barry Distinguished Professor and Head of the Vaccine and Immunisation Research Group at the Doherty Institute and Murdoch Children's Research Institute. Terry is a paediatrician and clinical epidemiologist, has been involved in immunisations and clinical trials of new vaccines for decades, and last year led the phase 1 trial of 2 novel Melbourne-made SARS-CoV-2 vaccines as a 4th dose booster. And Professor Paul Young is kindly joining us from sunny Queensland. Professor Young is a Professor of Virology. He's currently seconded to research development in the Deputy Vice-Chancellor's Office of Research and Innovation at the University of Queensland in Brisbane. And with a large team at UQ, Paul led an Australian consortium that developed a COVID-19 vaccine in 2020. Many of us followed this with great interest. The vaccine candidate was taken through to clinical trials and unfortunately didn't progress due to the cross-reactivity with HIV testing. And as noted at the time, Paul and his team have been working on re-engineering the technology, and that work is now progressing. So I will start with Professor Dame Sarah. The first person received the Oxford vaccine on 4th of January 2021. I'm sure you remember the day well. less than a year after the COVID pandemic had been declared. It was an extraordinary moment, I think, for many of us around the world, and I think it was the first person to receive any vaccination, but you might correct me on that. What was the moment like for you?
Well, it wasn't the first vaccination in the rollout.
mRNA was earlier.
mRNA was, but that was licensed. So the MHRA were very quick to license vaccines, and actually some vaccinations happened in December with mRNA vaccines, or one of them. But they licensed our vaccine, Vaxzevria is the name that it was given. They licensed it on the 30th of December, and the rollout started in January. So that was a culmination of a really, really tumultuous year of work by an ever-growing and very, very hard-working team. And it was fantastic to reach that point where we could finally start to get people vaccinated and start to get them protected. For me personally, it was quite frustrating because I wasn't able to go and see what was happening. We were still working under very strict social distancing. So I actually met Fergus Walsh, the health reporter from the BBC recently, and he said he felt very honoured to actually be present in the room when the vaccination was given, because it was literally him, his cameraman, the volunteer, so the person being vaccinated and the person giving the vaccination. And those were the only people who were present. But obviously, because it was filmed, we were able to share that with many, many people. But those of us who had worked on the development of the vaccines had to look on the screen and watch the news that evening to see what had been going on, because we weren't allowed to be there in person.
Did you crack a bottle of champagne that night with your family?
There was a day where we had champagne in the lab, socially distanced. And I think that was possibly on the day the vaccine got licensed rather than the first vaccination was given.
All right.
And it was less of an exciting event than we'd really all anticipated, because we had to collect our glasses and go back to our own computers. and it wasn't an amazing celebration, so we didn't repeat that experience.
Right, no secret parties at Oxford University, as were happening elsewhere in Britain. Sorry, Vaxzevria doesn't stick, and I keep on defaulting to AstraZeneca, but we should stick to the trade name. All of us watched this in great awe around the world. Or each of us were facing different challenges throughout the pandemic. How did you manage to move at such speed as an academic institution?
This comes out of more than 25 years of vaccine research, and not just immunology research in the labs, but actually vaccine manufacturing, vaccine clinical trials. We'd done 12 clinical trials with Challets 1 vector vaccines prior to April 2020, when we started the COVID vaccine trials. So we had all the teams in place, we had the expertise, we had the experience of working with the regulatory and ethical authorities. We'd done all of this before in terms of at least getting into a phase 1 trial. We went further in 2020 into phase 3 and expanding into other countries very quickly. But certainly for the starting the phase 1, it was a question of doing what the team knew how to do, but working out the fastest way we could do it, what had to be done before the next thing could be done, looking many steps ahead, and just getting everything off the ground as quickly as possible. And
what were the key challenges for you at that time?
In the beginning, it was getting funding for this work was a huge challenge. I had a little bit of funding, research funding, that I could use to just kickstart the process in the lab. But once we get to manufacturing the vaccine for clinical trials, that's an expensive process. And it was a constant problem through the spring of 2020, trying to find out how we were going to pay for the work that we felt that we really had to do, until we got to the point later on in April where the UK Vaccines Task Force was formed. And that was set up to assess all the different technologies that were being used to create vaccines and to test them, and to provide whatever funding was necessary for multiple different technologies owned by different people, with the aim of getting at least one of them through to licensure in the shortest possible time. So once that was set up and funding came through from them, we were really able to concentrate more on doing the work instead of worrying about where the money was going to come from.
And finally, what other diseases are you currently working on for which this technology will be applied?
So I'm concentrating on outbreak pathogens, along with some of my other colleagues at the University of Oxford. We've just started the third phase 1 trial of our Middle East Respiratory Syndrome virus vaccine. We are waiting to get approval to start a vaccine trial for Nipah virus, and that's a virus that's currently causing an outbreak in Kerala in India. We have a Lassa vaccine that's now in manufacturing that will be going into clinical development next year. And working with other colleagues on Crimean Congo hemorrhagic fever, Rift Valley fever virus. So all of the viruses on the WHO list that we know about that cause outbreaks that we don't yet have vaccines for, we're planning to work on all of those, thinking about how we respond to unknown viruses in the future by being as prepared as possible, and also making sure that we've looked at all the different families of viruses that are likely to cause outbreaks and make sure we're working on at least one example from each of those families.
Terry, I might move to you. Last year, you led the phase 1 clinical trial of 2 Melbourne-made COVID vaccines, one by the Doherty Institute and the other from the Monash Institute of Pharmaceutical Sciences. Can you briefly describe the technologies behind those candidates and why did we need another vaccine at that point in the pandemic?
Sure. We'd actually started well before that. In fact, we started early in 2020 in preparing one of those 2 vaccines that you just mentioned, a recombinant protein vaccine, based on not the whole of the spike protein, but on the tip of the spike, which is called the receptor binding domain of the spike protein. And there are reasons why focusing the immune response on just that tip may end up being a more efficient way of going in the medium and long term. So that's one of the answers to the question, why do we need different or new vaccines? But certainly at the time we started, there were no vaccines that were licensed. Sarah was starting their work. They were well ahead of what we were doing, of course. But nonetheless, at that time, we didn't know whether any vaccine was going to work. So like many other people around the world, that pipeline was being filled with potential candidates, and some proceeded very, very rapidly, including the AstraZeneca vaccine, those made by Pfizer and also by Moderna, similarly proceeded rapidly, and also Janssen, who made another vaccine somewhat similar to the AstraZeneca Oxford ChAdOx vaccine. Secondly, the mRNA partnership, mRNA candidate, which is the second of the 2 vaccines from our Monash colleagues from the Monash Institute of Pharmaceutical Sciences, was also similarly targeted at just the tip of the spike protein. And we had planned from the very beginning to actually have a head-to-head comparison of these 2 quite different candidates. Again, at the time this all started, no one knew that an mRNA vaccine was actually going to work for sure. And certainly whether it would be better or different to a more conventional approach or a modern conventional approach with what's called a recombinant protein vaccine. So that was the setup for doing this. And we actually started enrolling last year and finished the follow-up of those enrolled subjects for our phase 1 study in April of this year. And we're now just looking at the final data from that follow-up over the full 6 months of this phase 1 study. So we've had very, very good results from both these candidates. Where they will go in the future remains to be seen in terms of further development. But it was an outstanding success, really, in terms of the technology that was used and the capacity to actually make this happen here. Unlike Oxford, where Sarah has been doing preparatory work against some of these high lethality, more unusual diseases, which we don't have vaccines for, such as Ebola and the other vaccines targets that Sarah's just mentioned. So we really had a standing start in a sense. We had done many vaccine clinical trials before over the last 20 or 30 years, but more conventional vaccines with pipelines that might have been lasting 10 or 15 years to get to the point where they were going to human studies, both in children and also in adults. But really to start with a candidate early in 2020 and get it to phase 1 now completed itself is a remarkable feat. It's nothing, of course, in comparison to what Sarah was able to achieve in Oxford, and also with the funding and also the industry partnerships and contributions that you were actually able to benefit from, some of which I think were really exemplary ways in which corporate behaviour demonstrated a high degree of public good across the pandemic. And certainly the AstraZeneca partnership is the best example of that I've ever seen.
Are you able to briefly summarise the main findings from the trial?
Again, this is a phase 1 study. So phase 1 studies are generally to show, first of all, that a new vaccine is safe. And the summary is it's safe on the basis of the 76 subjects who were studied in this study, 12 of whom got placebo, so they just received water. Unknown to them or to us what they were receiving. It was a blinded study, of course. And the other subjects in the study, 32 who received the mRNA vaccine and 32 who received the recombinant protein at different dose levels. Similarly, no evidence that there was any issue with more reactions from the vaccines, and certainly nothing dramatic or unexpected. It's a small number, and this is how all phase 1s start. Secondly, in terms of the immune responses, to how well did they work? The study doesn't have enough subjects to give you a very fine-tuned, precise answer, but it gives a rough idea. And the rough idea was that both the candidates worked very well against the target receptor binding domain of the variant that was studied with these vaccines, and also a high level of cross-protection against Omicron and the other subvariants of Omicron that have subsequently evolved. At the time, no one knew what was going to happen with this virus mutation that we now, it's actually become almost part of common knowledge that people understand that these viruses are mutating very quickly. So anyway, the result is for both vaccines, very, very encouraging results against the targeted variant and also against other variants which have subsequently evolved.
And where to next for these vaccines?
Well, that's a good question. So we are talking with, and I can't give detail on this, but we are talking to one major international vaccine manufacturer about potentially one of these two vaccines, the recombinant protein, the one that's come from Doherty, potentially going into combination vaccines with other targets of commonly used vaccines, where COVID vaccines and other types of respiratory viruses are being immunized for in the future. mRNA vaccine, I can't speak for the Monash group and where they're going with that, but we're a university, we're not a commercial organization. We don't believe that it's appropriate that we should be taking other, except in exceptional circumstances, such as Sarah had to deal with the pandemic. And only then, as Sarah will explain, with the benefit of the resources and other infrastructure that a big company like AstraZeneca had, to go into to a full phase 2 or phase 3 study really needs a major manufacturer. And Paul's going to speak in a minute, and he'll talk about how that pathway was one that they'd stepped onto with their candidate as well.
Thank you. And now to Paul. Paul, the eyes of Australia were very much on the University of Queensland, and you personally, no pressure, at the start of the pandemic. And we know you were leading a very large team, not only in Queensland, but in collaboration with other groups around the country. And we had that disappointing moment when, unfortunately, the clinical trial needed to be abandoned due to diagnostic interference with HIV testing. A great disappointment for you and for many of us around the country. But at the time, you said you would go back to the drawing board and re-engineer the vaccine platform. So how is that work progressing?
Yeah, thanks. Thanks, Sharon. I should also acknowledge the traditional owners of the lands on which I'm currently sitting at the moment. That's the Turrbal and Yagara people, and pay my respects to elders past, present and emerging. It was certainly a challenging time, the end of 2020, when we had to make that announcement. It was a big call to say we're just going to pick ourselves up off the floor and re-engineer and move forward. But that's exactly what we did. I think we took a week off after 24/7 for a full year, but we did take a week off, gave the team a week off, and came back and redesigned, and we were back fairly quickly. And as has been announced previously, that's been somewhat successful, well, quite successful in terms of finding an alternative. The structural motif that we use that underlies the platform is seen across a wide array of different proteins, and we were able to choose one from many that works really well. And we were able to take that COVID vaccine back into clinical trial just earlier this year, and that was with CEPI funded support, and we'll be able to tell more about that in the future. But we were able to sort out a number of the issues that we dealt with during 2020, like, for example, downstream processing, all of the issues of manufacturing that a humble research scientist don't really think about until you get into the weeds of actually producing something at scale. So it's progressing well.
And I'm sure you and also your team have thought deeply about the challenges of advancing a candidate vaccine at pace. And I think all of us learned in the pandemic that Australia needs that sovereign capability, not only for our country, but also for the region in which we live in. Can you share some of your key insights about what the barriers are in Australia and what we can do better as a country?
Yeah, I think, let me go back to early 2020. We have a very similar story to Sarah in that regard, and that is we'd had a long history of work, not decades, but about 8 years of work had gone into our clamp technology. But the pivotal moment for us came when we were successful in being awarded a CEPI grant, a Coalition for Epidemic Preparedness Innovations grant, which began in 2019. And that was with significant funding, $15 million over 3 years to develop the platform technology itself. And one of the targets or candidates we chose was Middle East Respiratory Syndrome virus, so MERS, another coronavirus. So we'd had a year where we'd been specifically working on that particular target. But not only that, and I think this comes to your question, we had significant funding to do it. And that meant we were also able to reach out and support a wider team. And that included people at the Doherty and other academic organizations, and also into some NGOs, and ultimately into CSL, who agreed to partner with us early in 2020. And so to me, it's about scale. And what we need to be thinking of in Australia, I know we're going through a review process at the moment of MRFF and NHMRC funding and how that might be better organised and distributed. But what we really should be talking in addition about is an increase in government funding into the R&D sector more broadly. We're sitting at 1.65% of GDP at the moment, which is in the bottom third of OECD countries, well behind the UK. And we need to be shifting that up to at least, I would have thought, 3% or so. And with that, hopefully that would engender that broader national engagement and collaborative work. That was probably the biggest component of 2020 that I took out of the experience, and that was the power of partnerships. That broader national perspective, everybody keen to be working with each other with a single directive was really empowering. I guess the other things that I saw in terms of taking me out of my comfort zone, the whole team out of their comfort zone of moving a research project into a translational development pipeline, were all of the gaps that became apparent in that pipeline here in Australia that we just don't have. Money can get you through quite a lot of it through commercial organisations and funding them to do pieces of the work. But I think what we need to be thinking seriously about in Australia is developing capabilities, not just in the final end manufacturing space, which is also very important, but also upstream development phases. I'm thinking of one in particular, which is the biggest hurdle for most innovation coming out of research and development environment, and that is getting into that translation pipeline, getting a product that can actually go into phase 1 clinical trials. And it's an extraordinary complex process. You need a lot of expertise around regulatory environment, the GMP manufacturing of products and so on. But you also need to be able to make that product in the early stages. And that's a very expensive prospect and often a too big a hurdle to cross. So I think what we could be doing in Australia is really focusing to some degree on developing that early phase, small scale manufacture that can go into phase 1 clinical trials. We have a huge advantage in this country over any other country in the world. We have a particular pathway into phase 1 that does not necessarily require GMP material. And so the cost imperative is perhaps a little lower for us, but we're still not in the category of being able to make that material, that clinical trials notification pathway, the CTN pathway into clinical trials. If we develop some of that expertise and also link up our researchers with industry and the industry expertise around product development, I think those are the things we need to be focusing on.
Great. Well, thank you to each of our speakers, not just for the incredible work that they did throughout the pandemic, but for sharing their insights with us now. Very important to reflect on where we did well and where there are gaps and how we can address them going forward. Just as a scientific community, independently of any external review, I'm going to open up to questions. Please post your questions online, or if you're in the auditorium, place your hand up, and the auditorium gets first preference. And please introduce yourself and tell us who you'd like to direct your question to.
Hello, I'm Jamie Miller from Biotech Daily, and I just had a quick question for Dame Gilbert. How exactly did you secure the funding from AstraZeneca, and what would collaboration over not something that was necessarily a pandemic, but just any other even rare disease look like for you from a commercial perspective?
So what actually happened was that we formed a partnership with AstraZeneca, and that was something that was negotiated by others in the university, not myself, in the early months of 2020. And AstraZeneca agreed to work with us to take on the manufacturing of the vaccine, which we'd started for our first clinical trial, but we're going to need a lot more of it for the full clinical development and then for commercial supply after that. And they worked with us. So they didn't fund us directly. We had funding from the UK Vaccines Task Force, but what they did was set up a fantastic manufacturing network. They took our vaccine technology, they understood how to make it, they then transferred it to 25 different manufacturing sites worldwide, one of them in Australia at CSL, and got all of these manufacturing sites up and running and making essentially the same product so that the regulators would recognize it the same product. That took an enormous amount of effort from the company, very, very many hours to work with each. First of all, to identify the appropriate manufacturing sites, and then to work with them to transfer the technology and to make sure that everything was going as it should. They also ran some clinical trials themselves. They looked at what we were doing in our clinical development and said the best thing, because we need speed, is for you to carry on. We won't interfere, but they ran some separate trials in different countries. And they really were a great partner to work with in this crisis situation where we had to get things moving really quickly. They not only use their own expertise, but they allowed us to use our expertise, which is really important in a partnership.
Great, thank you.
Yeah, and just to follow up on that, what do you think collaboration amongst smaller biotechs would look like if they were, say, developing two diagnostic tools or two softwares, medical softwares, or drugs that were doing the same thing? Do you think that in some way the money kind of hinders the ability for companies to collaborate and develop? Or
so I'm very much in favour of partnerships between all the different scales of industry and academia. We do have to think about the funding, though. You know, somebody has to pay for the university research. And the staff that need to be employed there. So, and there are different mechanisms of doing this. One way of doing this is I work with a vaccine manufacturing research network in the UK. It's called VaxHub, and we encourage new partnerships, and we get money from research councils that allowed us to provide small amounts of seed funding to start up new collaborations. And that's often a good way of getting a new partnership underway.
Thank you very much.
Any other questions online?
We do indeed. The first question is for Dame Sarah. This is from Jason Gale from Bloomberg. I understand the chimpanzee adenovirus vector vaccine platform is being used for other immunisations, a shingles vaccine in China, for example. In COVID, the adenovector vaccines were associated with rare blood clots. Does the same risk carry over for other vaccines using the same platform? And have you been able to do anything with the platform to mitigate this risk?
So the difficulty with extremely rare adverse events is understanding what the causative mechanism behind those is. And we haven't fully got to the bottom of that yet. So we can't really answer the question about will it be seen with other vaccine antigens using the same platform? Will it be used in will it be seen with other adenovirus vectored vaccines? And we are working towards trying to understand what is behind this. I think it's going to be quite a long process, frankly. It's very difficult to investigate things that are incredibly rare. And when we saw rare adverse events against narcolepsy with flu vaccines in 2009, it actually took quite a number of years before that was fully understood. It's a very challenging thing to do. We're working on it, but it will, I think, be some time before we have a definitive answer.
Thank you. We've got another question. This is to all of the panelists. You might want to take a crack at answering this. This is from Henrietta Cook from The Age. What might the next pandemic look like, and how exactly are we preparing for it in terms of vaccine development?
Maybe I'll get Paul to answer that one to start with. Paul, are you happy to take that?
Yeah, I'll just grab my crystal ball it's a challenging question. What might it look like? I mean, what we've seen over the last 10, 20 years is a large number of emerging diseases that have manifested as local epidemics, epidemics that have spread to limited areas to the pandemic we've just been through. So it's very hard to know exactly what that's going to look like. Respiratory pathogens are the ones that tend to spread out in a pandemic environment more than others. So we're focusing on those. I think what has been realised, and this I have to give some credit here to the Coalition for Epidemic Preparedness Innovations, whose existence really came about following the Ebola outbreak of 2014, 15 and 16. And WHO's somewhat reactive response to that. WHO was saying, well, we do need to be more proactive. How are we going to do this? And CEPI was set up in part to fund research at a scale that would allow both the academic and small biotech and industry sectors to think about developing potentially rare use vaccines. So that work began in 2018, 2019, and has only been sped by the pandemic. So a large number of academic environments, but also industry now are playing in this space in terms of trying to develop these sorts of vaccines. So I think, and on top of that, each and every country is developing the local sovereign capacity, as we discussed back in 2020. It was a catchphrase that was often thrown around about our limited capacity here in Australia. We're starting to think about how we actually build that capacity more into the country's capacity. So I guess two things: that we're getting more and broader engagement by industry in focus on potential pandemic type agents and the development of both vaccines and therapeutics to combat those as a toolkit or even a stockpile for future potential emergence, but also locally developing in each country the infrastructure that will be able to move swiftly in developing some of those. So I think we are progressing. What it actually looks like, it will happen. We can guarantee that. Exactly whether it's a pandemic of the scale we've currently seen or a smaller spot-like epidemic, we yet to see.
Any further comments, Terry?
I'd just add that anyone who says that they do know, don't believe them. It's a species jump from animals in particular into humans that we're all worried about, that can't be predicted with any certainty at all. In terms of the statistical likelihood of previous pandemics, influenza remains the one that's most likely to be amongst the ones that will come in the future. We've had different cycles of influenza pandemics of different severity. People will remember the last was in 2009. But you will have heard the term being used by some of us today, platforms. So generally CEPI and other organisations and universities and Sarah's group in particular are trying to develop a type of vaccine that could be adapted very quickly to an unexpected virus that might come from a group of similar or related viruses. So rather than having to start from scratch, very quickly adapt, as Sarah did with her coronavirus, because they were already studying a different type of coronavirus, this MERS one that Paul also had mentioned, that was readily adaptable to the platform, and also was the same family of viruses. So that really made it very possible to do this very, very quickly. But so that so-called pandemic preparedness and having those sort of platforms available is really the most important insurance that we can do in the research sector to be prepared for whatever might unexpectedly turn up in the future.
We have another question from Jason Gale, which is similar. To what extent was the concept of Disease X helpful in developing countermeasures for COVID-19? And how much better prepared are we in 2023 for responding to a completely novel infectious threat that we were in 2019?
Might get Sarah to tackle that one.
So, as Paul said, the Ebola outbreak in 2014 and the really quite poor response to dealing with that did stimulate thinking around outbreak pathogens and looking at a list of pathogens that have been known to cause outbreaks previously and stimulating vaccine development for all of those vaccines on the list. And we still have a long way to go with that. We still don't, we are nowhere close to completing that task. We've hardly begun it, and we really need to return to that. And then following from that, there's this concept of disease X, that we might not, there might be an unknown pathogen causing an outbreak and we wouldn't know what it's going to be and how would we respond. So because we were using platform technologies, these very adaptable ways of making lots of different vaccines, we were using them for the outbreak pathogen vaccine development. We already had a head start in thinking about what we would do with disease X. But with outbreak pathogens, we'd been following the usual academic funding model of doing a little bit of work and then publishing the results and then trying to get money for the next stage. It was very, very slow. So in 2019, I'd started thinking about how would we do all this if we needed to do it quickly? How would we move from the idea through to actually having a vaccine in clinical trials in a really short space of time? Because we're going to have to do the same things technically, but we were going to have to work in a different way. I wasn't successful in getting that project funded, but at least it had meant that we'd done a lot of thinking about what would we need to do to respond fast if there was an outbreak with an unknown virus. And that's then what happened at the beginning of 2020. So we were able to put those plans into practice. So it's basically the same technology that we're using for outbreak pathogens, but a different way of approaching the research, which really minimizes the time that we spend before getting into clinical development.
And if I could just add to that, we're talking about vaccines, but vaccines are really only one arm of a response to an infectious disease. And in the therapeutic space, we took even double the time to get a direct-acting antiviral developed. And so the next big initiative here at the Doherty Institute is investing in a centre for pandemic therapeutics, the coming Global Centre for Pandemic Therapeutics, as a result of a gift from Jeff Cumming of $250 million over 20 years. Exactly the same principle of platforms that can rapidly adapt to a new disease, but for therapeutics, which hasn't really entered the mindset as much. We've used very similar technologies for therapeutics over the last decades, and we're hoping we can change that as well. Diagnostics, of course, we moved much quicker, but there's still lots of room to move to get that at speed.
Thanks, Sharon. We've got Henrietta Cook with another question. She's raised her hand. I'm going to allow her to talk. Okay.
Can you hear us?
Yes, we can hear you.
Hi, it's actually Aisha Dow here from the Age. I'm just sitting
Aisha.
here with Henrietta after coming out of my meeting. Thanks for allowing a question. Obviously, we've been talking about vaccine development and the speed of vaccine development. As an observer, it was incredibly impressive during the COVID pandemic to see how quickly vaccine candidates were developed. My question is whether that should come into our future planning, this expectation that we might be able to roll out vaccines potentially quicker than before. I guess in Australia's situation, we were of course able to kind of keep the borders closed and keep COVID out until vaccines came into Australia. I was wondering if that might be part of broader pandemic planning, that it might be possible to consider really trying to suppress a virus until vaccines could be rolled out.
So, Aisha, is the question, can we do it faster?
Should this influence, should our advancements in vaccine development influence our future pandemic planning? Might it be different from 2019 to now, based on I guess the development in advances in how we can roll out vaccines at speed?
Maybe Sarah, would you like to tackle that?
So there's a lot of thinking about how we can go faster. We still need to do a lot of research in this area, and really what we should be concentrating on is being as prepared as possible. The more that we can do before a new pandemic starts, then the better place we are. So we shouldn't just be thinking about getting ready to move quickly from a standing start. We should be thinking about how much of the work can be done in advance of a new outbreak starting or a new a new pandemic starting, because then we actually have a much better chance of achieving it. And there's many, many different factors to that. As Sharon was saying, it's not just about the vaccines, it's about diagnostics, about therapeutics, it's about the non-pharmaceutical interventions, and it's about the social science studies of how people respond to these interventions. Because I think, people have maybe changed their views since the pandemic as to what they will be prepared to do another time. And we need to kind of reevaluate how that all works and what research we should be doing in that area as well. So there's lots still to do. We've learned a lot in the last pandemic, but unfortunately, this has probably moved out of the public eye, but we need to keep it there, because there's a lot more that we need to do to keep countries protected for the future.
Aisha, did you have any follow-up questions?
We did have one further question, which is a fresh one, if that's okay.
Go for it.
Yeah. Obviously in Australia, we had in many ways a great situation in 2021. There was often we had days where there was often no cases of COVID in the country at all, outside hotel quarantine. But it made it a challenge when communicating the benefits of the vaccine to the public. And AstraZeneca in particular was a bit of a victim of that. And I guess the rare side effects, of course, we heard about were magnified in that environment. I guess the question is, what do you think the lessons are from the COVID pandemic around the communicating the risks and benefits of vaccines?
Maybe I'll flick that one to Terry, who in a prior life had a very prominent role in advising the government on vaccine policy. So Terry, what would you like to
Thanks,
comment on
Sharon. Well, there's a whole inquiry that's going to be looking at this in Australia shortly, but I think what we learned in Australia was that, as severe as it was, that lockdown did protect the lives of many. And in fact, life expectancy went up in Australia in the earlier part of COVID. There's a paper just published last week in the International Journal of Epidemiology from demographers who've actually shown that after all those restrictions and the lockdowns and so on were released, we did get some payback, if you like, of unimmunised individuals in particular who did die, and so there was some return to the previous levels of life expectancy. So, they're leaving aside all the contentious issues of compulsory or mandated vaccination, those sorts of things, which were very divisive at the time. There was an extraordinary level of compliance from the population with the public health approaches that we had. And there's no question that it saved many, many lives as a result. I think what we learned from all of that, though, was that in particular, you've got to have the public come along with you. This issue of communicating risks and benefits with the AstraZeneca vaccine was particularly difficult for us when the clots syndrome first appeared as a potential association with the AstraZeneca vaccine, which was very widely used here. I was vaccinated and Sharon was as well with that vaccine. All of a sudden, because we had no disease, because we had lockdowns, the issue about the relative risk and benefit of vaccination became very poignant for us. And it was a very hard thing to communicate. And yet, for someone like me, I've been involved in vaccination all my professional life. For children, we don't think twice, most of us don't think twice about vaccinating kids against diseases which have almost vanished, such as polio, for example. And those vaccines have risks associated with them. They're tiny, extraordinarily rare, but they are not risk. No vaccine is risk-free. And yet we do readily accept that. We don't go into complex arguments about risks and benefits with different scenarios of disease incidence, which we went through. And I think the public got very confused about all of that. So I do think we've got to find better ways of keeping that messaging simple and understanding where we're starting from and what we're doing in terms of protecting the future as opposed to protecting what might happen tomorrow.
Aisha, any further questions from you guys?
Sorry,
Oh, sorry. Are you there? Sorry, I accidentally muted you again.
Yeah, we're here.
Yes.
We just had one quick question around the COVID inquiry. Do you have any suggestions as to questions and things that you'd like it to look at in terms of the vaccine rollout and procurement?
I'll hand that to Paul. Any comments?
I'm going to pass that one straight back to Terry. I think that's Terry's space.
Yeah, well, yeah, and some of us were critical at the time about the government's, the previous government's handling of this, the procurement process and so forth. It's all water under the bridge now, and I think as a country we learned a lot about that, about having to be very rapid in our assessment, our engagement with international manufacturers. There was intense international competition for procurement of licensed vaccines, and it was an enormously difficult thing, particularly with the AstraZeneca vaccine. The issues around giving more people access to it, because it was introduced in a graded way to populations who were just to be more at risk initially before it was more generally available. About single dose versus 2 doses. Single dose works quite well with both the mRNA vaccines and also the adenovirus vectored vaccines, such as the Oxford vaccine. But at the time, we didn't have enough vaccine to protect all of the people we wanted to. We had huge demand for those vaccines to be made available. So the flexibility to change schedules, to vary times of administration became quite contentious at different times and very awkward. Eventually, the supply overcame that, and all of that became sort of a distant memory as vaccines were available to everyone. We moved into a different phase of having plenty of vaccine, but a reduction of enthusiasm to be vaccinated, particularly with boosters, because people could see that they felt that they weren't at risk. And that's one of the situations we're dealing with now, where we still have plenty of circulating virus, plenty of people still severely affected by the virus, and the need for boosters, especially in vulnerable populations, the elderly, those who have immune conditions which threaten their risk of infection. So a lot to learn, and I hope the inquiry will get into that. The political complexity that underlies all of this, in a way, is a pity, because those issues, I think, do distract from what the fundamental public health issues are.
Quick follow-up?
Yeah, sure.
So I'm not going to comment on what happened in Australia, because obviously I'm not from here and I'm not expert in the situation. But I think the question that we should be asking around the world is, how do we make sure we never get into this situation again?
No more questions that are coming through, so I'll hand it back over to you, Sharon, to close. Oh, and yep, no, Jason said no.
Okay.
Any further questions? No?
Great. Well, thank you very much for joining us in the auditorium and online. We look forward to doing an even better job if ever faced with another pandemic. Lots of lessons learned. And as you've heard, still lots of gaps to be filled, both in Australia and globally, in how to optimise our response. So thanks very much to our panellists, and have a nice day.